thanks to Deepak and the organizing team for giving me this platform to talk about some of the things that are going on in my laboratory with no promise of translation. So what I'm going to present to you today is fundamental biology of aging, and I will give you a lot of data. So I've been interested in the fundamental biology of aging for more than 20 years now. And from today's all the talks, you already know that the rate of aging is controlled by nutrition or food. And we know examples where excess nourishment accelerates aging. And on the other hand, we heard talks about dietary restriction, which lowers insulin signaling, lowers the mTOR signaling to activate autophagy and decrease protein synthesis, activate AMPK, and these are pro-longevity molecules and cytokines. So when we talk about nutrition, it's a complex mixture of macronutrients and micronutrients. And you'll be wondering, that most of our work and most of our literature on the basic idea of how nutrition regulates aging comes from macronutrients like fat, protein, and carbohydrates. On the other hand, how micronutrients regulate aging is a growing area of interest in the scientific community. And one of the micronutrients which I am interested in is vitamin B12, which is exclusively produced by bacteria, some of them. And it's also a classic example of interspecies interactions between the microbe and the host. And as you all know now that the microbes that reside inside an animal's body and outside are key resources for health and longevity. And they provide supplemental coding power to us. They code for proteins and metabolites that we cannot code for. They are rich sources of energy. They train our immune system, and most importantly, produce micronutrients like vitamin B12, which we cannot produce. And so it's quite natural that if there is an imbalance in the microbiome or the microbial population which resides inside us or outside, which is commonly called dysbiosis, it leads to many of the characteristic aging related diseases, like here neurodegenerative diseases, obesity, diabetes, and others. So this is a very complex biology, right? So you need simplified systems, and in our laboratory, we use a very simple and classical model system to study aging, which is called Cinorhabditis elegans. And we typically use it because it's having a short lifespan, it's quite transparent, easy to store, and most importantly, it is backed by powerful genetics. And having 70% conservation with the human genome, it's quite relevant to humans. And more importantly, it is a bacterivorous. So the bacteria serves both as its food and establishes a microbiota inside it. So you can modify or manipulate both the bacteria and the host and understand the interspecies interaction. Yes. So in the laboratory, typically we feed C. elegans with either of these two bacteria. One is called the OP50, it's a B strain of E. coli, or HT115, which is a K12 strain. And these worms seem to be very happy if they eat either of these bacteria. And they have internal homeostatic mechanisms which prevent random life history changes if you feed one bacteria and the other. And these are maintained by gene-diet interactions. Very important aspect of biology we have started to understand very recently. So these genes maintain the homeostatic state of these worms, and you would not see any changes unless these you have particular mutants which are either a mutation in NMAR1, which is a neuropeptide-like gene, in C. elegans, or in RICT1, which is a Richter or TORC2 ortholog, or in a proline dehydrogenase gene in the muscle. And typically in these mutant worms, they show altered life history traits or increased or decreased lifespan on one bacteria and not the other. And over the last several years, we got interested quite by chance on a serotonin kinase called FLUR4, which shows increased longevity and stress tolerance on particularly the HT115 bacteria, but not on OP50. So a long time ago, when Sonia Verma, who is now a faculty in CDRI Lucknow, joined my lab as a graduate student, quite serendipitously she found that this FLIR4 mutants have an exceedingly long life when they were grown on this bacteria, but on the OP50 bacteria, it had a normal lifespan. As I told you that the wild types do not really care whether you feed this bacteria or the other. Now, through some careful genetic analysis, she showed that in the FLUR4 mutant fed on this bacteria, there is activation of the P38 MAP kinase pathway, which is a conserved innate immune response pathway in C. elegans, and that leads to increased cytoprotective gene expression, several tolerance, including osmotolerance, and thereby increasing lifespan. And she could visualize this entire process using a simple biomarker, which is one of these cytoprotective gene, promoter fused to GFP. And as you can see, only when the mutant worms are grown on this bacteria, you see the expression of this transgene and not on the OP50 bacteria. So throughout my talk, I'm going to show you only this glow, and you have to correlate this with increased longevity and stress tolerance. That will make both our lives easier. So when Tripti Nair joined my lab, she started understanding what is the difference between two bacteria, and she found that this HT115 bacteria has intrinsically higher B12 content compared to OP50. And when the worms are fed B12, and as you would know that the worms do not make B12, they depend on E. coli, which also do not make B12, and E. coli now has non-specific B12 pumps which pick up B12 from the media and provide it to the worms. When we feed the worms with high B12 diet, the internal concentration of B12 in these worms also increase. And what she showed very nicely is that when she supplemented the low B12 OP50 with increasing concentrations of B12, she got back the gene expression. And of course, the lifespan on the low B12 diet can be rescued by supplementation. So from now on, I'm just going to show you this data. So next, so what does B12 do? So B12 is required by two enzymes in the system. One is methionine synthase, which I'm going to talk about today. And the other one is a mutase, which converts methyl malonyl CoA to succinyl CoA. That's another story which I'm not going to talk. So B12 is required by methionine synthase to convert homocysteine into methionine. And this enzyme integrates two very important pathways, the methionine cycle and the folate cycle, and this constitutes the one-carbon metabolism of an organism. So to summarize the two work that I just presented, what we find is that in the FLUR4 mutant, the one-carbon cycle flux is increased, and that leads to an increase in the signaling of the P38 MAP kinase pathway in the mutant, and that leads to increased cytoprotection, stress tolerance, and longevity. Now, when Shabnam Sahin Rahman joined my lab, she asked the simple question, is this metabolic pathway and this signal transduction pathway temporarily separated in the worms? So how did she address this question? So as I told you, C. elegans is a genetically amenable system, and you can put in or knock out gene at ease. So what she did is she made a FLUR4 and METR1, methionine synthase double knockout, and put back this metabolic pathway only in the intestine or in the neuron. And what she found, very interestingly, is that only when she rescued the metabolic pathway in the neurons did she get back the gene expression and longevity, but not in the intestinal cells. And when she rescued this back into the intestine, into the neuron, these worms now started responding to B12. As you can see, on low B12 diet, it doesn't glow, but when the neuronally rescued worms are fed high B12 diet, they start glowing. So the next question was, where does P38 MAP kinase pathway work? And in a related experiment to what I just showed, what she did is knock down the expression of the gene either in the intestine or in the neurons. And just simply by knocking down the gene in the intestine, she could, by knocking down one of these upstream kinases of P38 MAP kinase, she could reduce the gene expression and the longevity significantly. So to summarize, what we have is the metabolic pathway downstream of the fluor4 functions in the neuron, and the signal transduction pathway functions in the intestine. So she asked what could be the mechanism of the neuron-gut signaling. So to cut the long story short, she figured out that both neuropeptide and neurotransmitter signaling were involved, and the neurotransmitter turned out to be serotonin. So if she makes a fluor4 TPH1 and double mutant, where TPH1 is the rate-limiting step in serotonin biosynthesis, the gene expression goes down, so does longevity, and simply adding serotonin from outside could rescue this. So next she showed that when the mutant worms are fed high B12 diet, there is a transcriptional upregulation of the TPH1 gene, and that leads to increased serotonin biosynthesis in the serotonergic neurons. So next she asked, where is serotonin working? It could work practically here, here, or here. And one of the experiments she did, among many, is to knock out the metabolic pathway in the neuron and then supplement serotonin. And she found that that could rescue the phenotype, placing serotonin downstream of the metabolic pathway. So finally, she figured out what are the serotonergic neurons which are involved, and out of the 6 pairs of serotonergic neurons, she found only rescuing the metabolic pathway in a pair of serotonergic neuron was sufficient to to complete the entire signaling process and bring back longevity as well as gene expression and stress tolerance. So it says that B12 is actually working only in 2 serotonergic neurons in the head region of the worms, which we call brain, and that can regulate systemic aging by activating P38 in the intestine. So if you have serotonin, you need to find the receptor for serotonin, for which she systemically knocked out the serotonin receptors, and it turned out to be MOD1, which is unfortunately not expressed in the intestine, which would have made our life quite easy, but is expressed only in some of the interneurons. And if you knock out MOD1, you suppress the gene expression and longevity, and these worms now become recalcitrant to serotonin. Yeah, and it becomes recalcitrant to serotonin. Serotonin doesn't work. So once we have the MOD1, as I told you, MOD1 doesn't express in the intestine, so we did not have the connecting loop. So we went back, and again, to cut a long story short, figured out that this is bridged by a neuropeptide, which is a glycoprotein hormone in C. elegans called fluor2. And this fluor2, when you knock fluor2 out in the fluor4 mutant, you take away the gene expression as well as longevity, and again, it becomes recalcitrant to serotonin. So now the important question is that is fluor2 secreted by the same neuron which receives the serotonin signal? So for that, what Shabnam did is she rescued the expression of fluor2 in the mutant using a MOD1 promoter, putting fluor2 back only in the interneuron which expresses MOD1. And as a result, she could get back the expression of transgene, the GFP, as well as lifespan. She went ahead to also identify the receptor of fluor2 in the intestine, which happens to be a follicle stimulating hormone receptor like protein in C. elegans, and as expected, it suppressed the gene expression and longevity, and this also is recalcitrant to serotonin. So together, this is the model that I want to end my talk with, that in the fluor4 mutant, which has a long life on high B12 diet, the methionine cycle and the one-carbon metabolism is active only in a pair of serotonergic neurons and leads to increased flux through the pathway which we had measured by metabolomics. And this leads to transcriptional activation of TPH1 and an increased serotonin, which is received by an interneuron having MOD1 receptor. And this MOD1 containing interneuron expresses and secretes this fluid-2 neuropeptide, which binds to the cognate receptor in the intestine, and through a mechanism which I have not shown, leads to phase transition of this SARM domain containing toward interleukin-like receptor 1 protein on the lysosome related organelles in C. elegans, and this leads to activation of the downstream P38 MAP kinase pathway. And then this whole pathway is functional only when the serotonin kinase is mutated, and these are grown on high B12 diet. So very interesting, though, although it controls longevity and stress response, data that I didn't show, and it's, I think, more interesting to many of the neurobiologists, is this exact circuitry controls behavior of the worms. So now these worms are prefer high B12 diet if they are given a dietary choice. So we are working currently on identifying the targets of this serotonin kinase, and that is turning out to be a very exciting project. With this, I thank you and introduce you to the members of my laboratory. And this was mainly carried out by Shabnam Sahin Rahman with Shreya Bhattacharya, and is now being taken forward by Prerana Ghosh. Thank you.
Thank you, Arnab, for timely talk. So, a couple of questions. So while asking, so the pathway what you're identifying is a alternative pathway, or is a measure can come because your knockouts worms are supplementing by B12. So in a natural scenario, how B12 act onto this is a negative regulator or positive feedback?
I didn't get your question.
So the B12 in a wild type C. elegans will act as a positive regulator or negative regulator for the entire pathway.
So it would act as a positive regulator, right? And this gene is preventing the increase in flux in the neurons.
I got now.
Did you see any decreased reproductive fitness for this increased lifespan in the worms?
Good question. That we did a long time ago, and there is a decrease in reproductive fitness, particularly in the number of eggs. As in all long-lived species, mostly mutants in the lab, there is less reproductive fidelity. If not reproductive fidelity, the number of progeny is less, although reproductive fidelity is very high. The quality of progeny is very high, the number is less.
Thank you.
So, Arnab, how did you identify B12 is a causative for your?
Oh, that took us a while.
Well,
yeah. So we looked at many other metabolites. In fact, I didn't say. It's actually B12. It's a methionine which is more important, and it's sourced from the bacteria, right? So bacteria also uses the same B12 to make methionine, and both these micronutrients are now important for the longevity.
thank you. Thanks a lot.